Hey guys. So in medieval England, when criminals were trying to run away from people, they would hide in churches, and the church would give them protection for 40 days. So the townspeople would surround the church and wait for the criminal to come out, so that they would be able to catch this person, um, so that they wouldn't sneak out in those 40 days. And this kind of resembles what we're going to talk about, which is malaria. So let's say that someone got bitten by a mosquito, and this mosquito has to be a female. And it's like the Anopheles mosquito or something. And it releases these sporozoites. So they're called sporozoites, which are a form of Plasmodium parasite, which causes malaria. So these sporozoites go to the liver. Now, why specifically the liver? So in immunology, you have to question everything. So why the liver specifically? It's because one, the cells, the hepatocytes, there's a lot of metabolism going on in the liver. There's a lot of protein synthesis because it needs a lot of enzymes for those enzymes to be used in like glycogen or like metabolism, biochemistry. So there's a lot of protein synthesis happening. There's a lot of replication happening. So it's like a convenient place for the sporozoites to grow. And also the capillaries in the liver have fenestrations, and so it's easier for those sporozoites to go into the hepatocytes. And also the liver has a lot of glucose and a lot of lipids and glycogen in like it has a lot of like food storage, so that's very convenient for the sporozoites. And the sporozoites also express two specific proteins called CSP and TRAP. I'll put the long versions of these proteins or these receptors in the description below. And they bind to these receptors on the hepatocytes called heparin sulfate proteoglycans, or HSPGs, and that allows these sporozoites to enter the hepatocytes. Okay, so now let's say we're in a hepatocyte, and this is a hepatocyte, and there's all these sporozoites that are in the hepatocyte, and at the moment they're babies. Okay, so sporozoites are like the baby form. Okay, and as they take up all the hepatocytes nutrients, they grow up into merozoites. So merozoites are now larger versions. So when there's a lot of merozoites in a hepatocyte, then that hepatocyte is called a schizon. I know it's a weird name, but that's what it's called. Okay? But the problem is that hepatocytes are checked by the immune system, which is like the police of the body. And so it's going to check these cells and say, there's a problem, you guys got to leave, or like the cell has to die. And that's because hepatocytes express MHCs. And so the immune system can recognize if the hepatocyte is infected and therefore do something about it. So now these merozoites have to go somewhere else. And so they're going to look for a new place to live. Now they're looking for a place that has no protection but has a lot of resources. And so they find the red blood cells. The red blood cells have no immune protection because they don't express MHCs, and they have a lot of hemoglobin and oxygen. The merozoites are kind of like a country or a people that we know that look for other people's land and that land is not protected by anyone, like by any other country, and has a lot of natural resources. If you know, you know. Okay, so so they go to the red blood cell and they eat up all of its oxygen and its hemoglobin. Now, hemoglobin, as we know, is formed from heme and globin. Heme is toxic to the merozoites. So, the merozoites convert heme into hemozoin. Now, hemozoin is not toxic to the merozoites, but the hemozoin has a secret, and we're going to look at that secret in a bit. Okay, so now these merozoites eat up everything that's in this cell and kill everything inside until the red blood cell dies and bursts and releases all of the merozoites into the blood. And so the hemozoin that was formed is also released. Now, the secret of the hemozoin is that it likes to attach to parasitic DNA. It very strongly attaches to parasitic DNA from the merozoites. So so this complex, which is the hemozoin with the parasitic DNA, is going to be recognized by macrophages. These macrophages are going to phagocytose this thing, and it's going to activate TLR7 and 9. The parasitic DNA is going to activate the TLR7 and 9. Okay? And TLR7 and 9 will lead to an increase in IL-1 and TNF-alpha. And we know that these two interleukins lead to fever. And so that's how you're going to get fever. So there's one phase where all the merozoites are inside all the red blood cells, and so there's no fever. And then there's another phase where those red blood cells burst and all these merozoites get released, and that leads to fever. And so this is the normal fever pattern of any other infection. Like whenever you get sick, you get a fever for a while, and then the infection goes away, and then your fever goes away. But in malaria, you initially, when there's the merozoites, which are in the blood initially, just outside, not in the red blood cells, that leads to immune activation. But when they enter the red blood cells, the fever goes back down. And then after a while, when the red blood cells rupture, the fever goes back up. And that pattern keeps happening. And so this fever is periodic, or it's like at a specific time. So for different types of malaria, like Knowlesi is every 24 hours, and then Vivax Oval is for 48 hours, Malaria is 72, and then falciparum is like the worst one. It might be irregular, or it might be every 48 hours. Okay, now there's another problem with Plasmodium falciparum. So we said it was the worst one. So there's another problem with it. It has this protein called PfEMP. Okay, Plasmodium falciparum erythrocyte membrane protein. Okay, and it looks like this hook kind of thing. And the problem is that if a lot of infected red blood cells express this protein, then they hook together and form this aggregated red blood cell clump kind of thing. And that's not good, because they're now very large pieces in the blood, and that's going to cause like clogging in like very tiny capillaries, like the distal capillaries, like the ones in the middle of the brain, like the little capillaries that supply the internal capsule, for example, and that will lead to cerebral malaria, which is found in Plasmodium falciparum specifically. And then also we know that the kidney has glomeruli, which is like very, very tiny capillaries, and so these will block those little capillaries as well and cause nephropathy. And so here we can see, okay, nephropathy here, and then also because TNF alpha and interferon in blah, blah, IFN, okay, whatever. Okay, so these will be released, and TNF alpha induces nitric oxide. Nitric oxide leads to vasodilation, which we'll talk about in a bit. Okay, and also there will be brain damage because of those rillettes, or like those like rillettes are basically like clumps of red blood cells, and then like interferon gamma will lead to further activation of T cells. Okay, and then also, okay, so children who have polymorphisms where TNF alpha is like excessively produced, tend to have a worse prognosis. Okay, so let's discuss this whole like nitric oxide thing. So nitric oxide, we know, causes vasodilation. So let's say this this blood vessel gets vasodilated. So when there's vasodilation, the larger molecules go to the edge of the blood vessel, and the smaller molecules stay in the middle. And so because these aggregated red blood cell rillettes go to the edge of the blood vessel, there's not going to be enough red blood cells going to the brain or going to wherever it needs to go to. And so that will lead to further ischemia to those distal, to the areas that are supplied by these distal capillaries. And then also another goal of malaria is to prevent being recognized by the spleen, because yes, these red blood cells don't have MHCs, but the way the body knows whether this red blood cell is infected and whether they should do something about it is via the spleen. These red blood cells go to the spleen, and then if there's any issues with the red blood cell, then the macrophages in the spleen take care of those red blood cells by either killing them off or like their mechanisms. Okay, so by them being stuck to the edges over here and not being able to get to the spleen, the malaria basically checks off this goal of staying away from the spleen so that they don't get recognized. Okay, now there's an issue. There is the RBC issue, because on one hand there's a lot of hemolysis because the malaria is taking all of the red blood cells resources and killing them, and then on the other hand, whenever there's an infection, whenever there's high TNF alpha, the body is trying to produce more erythrocytes because they know there's an infection. So what happens is when there's high TNF alpha, it tells the bone to suppress erythrogenesis, or like the production of red blood cells, and increase the production of erythrocytes. And so on one hand, red blood cells are being broken down, and on another hand, they're not being produced as much either. So that's a problem, and that leads to anemia. Okay, so now, is there a way to fix this problem, or is there a way to for the immune system to find a way, like a way to do like Uno reverse on the malaria? So one, there's immune education. So if an infection infects you many times, your body will find ways to deal with it. So for example, if this is like your 5th time getting infected with malaria, you'll have memory T cells or memory B cells that will release a lot of IgGs that are specific to the malaria and like stop it from getting to the hepatocytes or getting to the red blood cells in the first place. Okay, and then like throughout the centuries and decades, I guess, in endemic areas there have been mutations which prevent malaria from infecting like those, like certain people, and so it's like congenital immunity. Okay, and one example is sickle cell. So sickle cell trait or sickle cell disease, what happens is, um, so so we know that the malaria, the plasmodium, it takes away oxygen in the red blood cell, right? So when in sickle cell, when the red blood cell gets deoxygenated, hemoglobin S aggregates, and when it aggregates, it does not allow the merozoites to digest that hemoglobin, and so when it can't digest it, خلاص, it dies. And if the hemoglobin S, like when it aggregates and it goes through the spleen recognizes, okay, it's aggregated, and so it destroys that red blood cell. So it destroys any infected red blood cell, which is good. And so they have in like immunity. And then it's a similar thing with thalassemia, where thalassemia, they have more hemoglobin F, and hemoglobin F holds on to oxygen very tightly, and so it does not allow the plasmodium or the merozoites to take that oxygen. And then similar with hemoglobin C, which is like another mutation. Now there's one called Duffy antigen. Okay, it used to be like some people had the Duffy antigen in their red blood cell, like in their hemoglobin or on their red blood cell. Now Vivax and Ovali express Duffy protein, and so if your red blood cells have Duffy protein, then that's considered a self protein. And so when these malaria, like when these plasmodium came into the body, the immune system thought it was self protein because it recognized Duffy as self protein. So it could not mount an adequate immune reaction because one of its proteins was considered self. Okay, and then also Duffy antigen helps the merozoites to enter the red blood cell. It's like, come and destroy me. Like that's what Duffy, like it's like, please come and destroy me. Okay, and so those people died. Okay, and so that's why I don't think there's anyone in the world who has like the Duffy antigen anymore, because they've been eradicated. They died. Okay, and yeah, that's it. Thank you to Dr. Farhan for teaching me and my batch. And then Janeways is a really good book that you can refer to. And then also thank you to ChatGPT for helping me understand and simplify this lecture. Okay, good luck.